Okay, so today we're going to start with what a neuron is within the brain and how it functions. So I'm going to start with first a basic overview. So first of all, you need to know what parts are on a neuron. So a neuron has a cell body, represented by this pumpkin. He's cute. And the nucleus, which is within the cell body, which is shown by this light. Now, attached to the cell body is these dendrites, which are here. And then attached to the cell body is the axon hillock, which connects the axon and the cell body. So the axon sends the signals to these receptors over here. Now you'll notice that there's spiders on the axon. Now the spiders represent the myelin sheath, which help to protect and also to send signals faster through the axon. The spaces between the spiders are called the nodes of Ranvier, so it's just the space between each myelin sheath. And that's your basic overview of a neuron. So what happens is, say you touch your arm or something. The dendrites will feel that action, or that they'll get that sense, and they will then, determined by what happens within the cell body, if it is an action potential and the signal does get sent through, it'll be sent down through the axon, which some say that it seems as if the signal is jumping between the myelin, but it's actually going through within the myelin. And it says, as I said earlier, the myelin acts as like a speed, helps to enhance speed for the signals, and they end up down into the receptors. Now that's the very basic overview. So let's go ahead and get a little bit deeper into what each part is. So this is the brain at resting potential. This is what this is what the cell, particularly the membrane, looks like at resting potential. Now the membrane is what makes up the outside of the cell. So it would be like the pumpkin's skin here. That's what these colors represent. This is the cell membrane, which is made of phospholipid bilayers. And this is the extracellular side of the cell. So this would be outside the pumpkin. And this is the intracellular side. So this would be inside the pumpkin towards the nucleus. So towards the light here. Now what happens is when the dendrites receive a signal and send it to the cell body, this is the blown up version of that. So this is what happens within the cell body to determine whether a signal gets sent down the axon or not. So this is what it starts out as. We have our lovely color-coded membrane here. So the two ions that we care about are the sodium and the potassium ions, which are both positively charged. Now within the membrane there are two other ions, which don't really matter for our purposes here, but I'll still show them. And that is the chloride and the proteins, which are both negatively charged. Now proteins, for most purposes for showing the membrane, are labeled as A-. And as you can see, there's also a chloride channel. But again, we're just going to ignore this side for right now. So right here we have potassium, and as you can see, I've color-coded for clarification. So the potassium ions have two channels, and the sodium ion has one channel. Now the channels are what allow the ions to go back and forth between the extracellular and intracellular side, which is what creates an action potential within the cell, which tells it to send a signal. So what the brain looks like at resting potential is here, and this is the graph. So the brain at resting potential starts at negative 70 millivolts. And this is just time across, showing you as time continues on. So say like when you touch your arm, the dendrites feel it, they send a signal to the cell body, and the sodium gate opens. Now when this gate opens, the ions start to flood in because they are excited to get away from all these negative ions over here, the chloride ions. So they flood in. And as they flood in, the cell increases to a negative 50 millivolts. So when it reaches negative 50 millivolts, this potassium gate, which is super, super duper slow, starts to open just a little bit. It's not nearly as fast as the sodium channel gate that opened automatically. So while that gate is opening, these sodium ions still continue to rush in. And as you can see, there's always a couple ions on either side left open. So, as this continues to rise, it peaks at a positive 30 millivolts. And that is when this sodium gate closes and this potassium gate finally opens. So then all these potassiums are super excited and they get to flood out into the cell. So when that happens, it causes the brain to dip, hits negative 50 millivolts, which means that this potassium gate slowly starts to close, and that these sodium gates switch, so that they end as they started from the beginning. So now, however though, with the potassium ions being on the outside, the cell continues to dip and reaches a negative 80 millivolts. So now, our brain, of course, doesn't want to be at negative 80 millivolts. It wants to be at negative 70, which is where it started at resting potential. So, that's where the pump comes in. It then takes 3 sodiums for every 2 potassiums and helps put the cell back on track. So, woo. Okay, however, we have a problem. These little potassium charges over here, since the pump doesn't work with a straight ratio, it's a 2 to 3 ratio, these potassiums sneak through this open channel, putting the brain back at negative 70. And so that is what happens within the cell body to determine whether a signal is sent or not down the axon. So in this case, obviously a signal was sent because the gates opened, and the channels and gates opened, allowing for the sodium ions to rush in and the potassium ions to rush out, and then for the pump to refilter them back to the original resting potential. Okay, time for step 3. So in step 3, we have the synapse. So this is a blown up version of the axon terminal shown from the cell earlier, the neuron earlier. So what happens here is that the signal gets sent down into the neuron transmitters, which is the signal, gets sent down into these handy dandy little vesicles, which then sneak through into the synaptic gap, which then in turn send all these little neuron transmitters into the synapse. So what happens is that some of these neurons will hit one of the receptors on the second neuron, causing it to open a channel, allowing a bunch of these neuron transmitters to sneak through, which then starts to signal the system all over again of the signal going to the cell body, going the decision factor at the axon hillock, sending down to the axon, into the axon terminals where this happens. So not all of the neuron transmitters are taken into the cell for the second signal. Some are taken into a reuptake channel where they are reuptaken, recycled, and reused for the cell again, the neuron again, which would be shown by here. Some are eaten by an enzyme and destroyed, which is shown here. So they just continually be getting broken down by this enzyme and then eventually just disappear. And others are taken out by this autoreceptor, which just shuts down the whole process, which is really important because if your brain doesn't stop, if you don't get rid of these extra neurotransmitters, it's just going to continuously be sending a signal, which then the neurons won't know when to send a signal that could be very important, or they will just kind of shut down a little bit. And that's basically what happens within a neuron. Okay, and now just a quick overview for how the neuron works again. So we have the cell body, the dendrites, and when a neurotransmitter hits the dendrites, that causes a signal to be sent into the membrane, which is our handy-dandy color here, right here, which is then used to show how the potassium and sodium ions in their channels work to change and to send signals within the neuron. When they're decided to be sent, that's in the axon hillock, the decision-making point. that connects the cell body to the axon. Here's the axon with the myelin sheath, which the myelin sheath works as an electrical charge insulator, so it keeps everything within the axon, it keeps it in there, and which also helps speed up the process. So that's why it seems like the transmitters are like jumping between the myelin sheaths, but they're actually not. They're just sliding, like being sucked right through. And then we have our neuron transmitters, which is where we have our handy dandy little synapse with the vesicles and the neuron transmitters that can either A, be sent to start another signal, B, be taken up into our reuptake channel. They can be eaten by enzymes or they can be shut down with the autoreceptor. And that is how the neuron works.